The cardiovascular system contributes to homeostasis of other body systems by transporting and distributing blood throughout the body. As that blood is traveling through the cardiovascular system, it's delivering materials like oxygen and nutrients and hormones, but also picking up waste products so that we can get rid of those by filtering the blood or get rid of CO2 by exhaling when that blood drops the CO2 off in the lungs. So the structures that are involved in moving blood throughout the body are called the blood vessels. These form a closed system of tubes that carry blood away from the heart, transport the blood out to body tissues, and then returns that blood back to the heart. So in this chapter, we're going to focus on the structure and function of the different types of blood vessels. We're going to also look at the forces involved in circulating blood throughout the body, and on the blood vessels that constitute some of the major circulatory routes. Now, the biggest part of learning the blood vessels you'll learn in lab, but we will look at some of the major vessels as we're talking about maintenance of blood pressure, and also just to sort of get an idea of the vessels and where they're located. The 5 main types of blood vessels in the human body are arteries, arterioles, capillaries, venules, and veins. Arteries carry blood away from the heart out to other organs. We have several different types of arteries that we'll look at, including large elastic arteries that leave the heart and then divide into smaller and smaller arteries that go out to body organs, and then the smaller arteries are going to give rise to even smaller arterioles. So artery, taking blood away, and oles, O-L-E-S, means small. So the arteries are then going to enter, or the arterioles will enter tissues, and then they're going to branch into teeny tiny little vessels that are called capillaries. So the capillaries have very thin walls, and they allow for exchange of substances between the blood and the body tissues. Groups of capillaries in a tissue are then going to merge to form tiny little small veins that we call venules, and then the venules will merge into larger and larger blood vessels called veins that carry blood from the tissues back into the heart. So 5 different types of vessels, but all of the blood vessels contain 3 layers or tunics composed of different tissues. So all the different covers or tunics have epithelial tissues and smooth muscle and elastic connective tissue, and an outer covering of connective tissue. So we'll begin by examining the 3 primary layers and start with the tunica interna, sometimes called the tunica intima. It is the closest to the blood and the most internal layer. So it forms the inner lining of the blood vessels and is in contact with the blood as it flows through the lumen or opening of the blood vessel. The inner part of the tunica interna is an endothelial layer, and it is also a very smooth, very slick layer that's continuous with the endocardial lining of the heart. Remember when we talked about the heart, I said the endocardium is continuous with the lining of the blood vessels. So now we're looking at the lining of the blood vessels, and it is continuous with the endocardial lining of the heart, right? So basically it's just the endothelial layer inside the blood vessels in the heart is one smooth continuous sheet. So this smooth surface facilitates efficient blood flow by reducing the friction that the blood might have as it's passing through those blood vessels. The middle layer is called the tunica media. Media is middle. So it's a muscular and connective tissue layer that has the greatest variation when we're looking at different types of blood vessels. In most vessels, though, it's a pretty thick layer comprised mostly of smooth muscle cells, but also with substantial amounts of elastic fibers. The primary role of the smooth muscle is going to allow the regulation of the diameter of the lumen. So the smooth muscle in a blood vessel encircles the blood vessel, very much like a ring encircles your finger. But imagine that the ring covers your entire finger rather than just one little area, right? Okay, so because of that circular pattern of the smooth muscle, the nervous system can stimulate that smooth muscle to contract and squeeze the vessel wall, and it narrows the lumen. So the diameter of the lumen becomes smaller, and that's what we call vasoconstriction, to constrict the vessel. There are other times when the sympathetic nervous system is going to is going to cause a vasodilation to occur. So vasodilation is when the diameter of the blood vessel gets larger, so those smooth muscle fibers relax. So the flow of the blood through the different parts of the body is going to be regulated by the extent of smooth muscle contraction or relaxation in the walls of particular vessels. And this, of course, is crucial in the regulation of blood pressure. In addition, as we've already seen when we talked about hemostasis, this smooth muscle is what's responsible for that vascular spasm, that first action that occurs in blood clotting. Remember, in vascular spasm is when the blood vessel constricts, and that helps to narrow the diameter of that vessel, and then, of course, that's going to limit the amount of blood flowing through that damaged vessel and being lost to the environment. The outermost layer of blood vessels is called the tunica externa. You may sometimes hear this called the tunica adventitia, and that's because it's composed of a lot of elastic and collagen fibers. So the tunica externa, very fibrous, is going to help anchor the blood vessel to the surrounding tissues. So here you can see a comparative diagram of an artery, a vein, and a capillary. And notice here with the artery, we of course have the tunica externa on the vein, the tunica externa. The tunica media in the artery is very substantial, very thick as compared to the tunica media in the vein. And then we have the tunica interna, the endothelial lining in the artery and in the vein. And the only difference you see with the tunica interna, of course, is the vein is going to have these little structures called valves that we'll look at when we're looking a little bit more detail about what the veins are doing. But the big difference here, notice you do have a bit thicker tunica externa on the arteries, but a much more thick tunica media. So those arteries are going to be able to withstand a lot more pressure. So when we begin talking about blood pressure, remember this. And here you see the capillary, or a structure of a capillary. And of course the tunics are much, much thinner here. You have a basement membrane and an endothelial lining only. And it's very important that the capillaries are much thinner, and that allows for substances to pass from the blood into the surrounding tissues. Now remember that our circulatory system made up of these blood vessels is a closed circuit. So blood doesn't stop anywhere along the way. It flows from the heart into arteries. So arteries begins with an A, takes blood away from the heart, and then that blood flows into smaller and smaller arterioles that are going to feed into capillary beds. So here we have the heart, large arteries, large arteries, large arteries, becoming smaller and smaller arterioles that then feed into capillary beds. So the capillaries are the smallest of the blood vessels, and that's where exchange takes place from the blood into the tissues and from the tissues back into the blood. The blood then flows into very small veins called venules, and those small veins merge to form larger and larger veins, which will eventually form the vena cava, which feed right back into the heart. So arteries take blood away from the heart, veins bring blood back into the heart. The wall of an artery has the three typical layers that we see in blood vessels, but it has a very thick muscle and elastic tunica media. So because of that very plentiful amount of elastic that's found in the tunica media, arteries have a very high compliance. That simply means that their walls can stretch easily or expand without tearing in response to small increases in pressure. Remember, arteries are taking blood away from their heart, so they're getting that blood when it's under its highest pressure, which is when the heart is going to contract and send blood out those ventricles, as we saw in the last unit. So we'll look at some different types of arteries. Elastic arteries are the largest arteries in the body, and they range from a large aorta about the size of a garden hose to some very, very small arteries, such as some of the arteries that go out to body tissues. Elastic arteries include the two major trunks that exit the heart, and you know already those are the aorta and the pulmonary trunk, and also the aorta's initial branches that come off at the top of the aorta, such as the brachiocephalic artery, the subclavian artery, and the carotid arteries, and also in the abdominopelvic cavity, the major branches are the iliac arteries. So all of those arteries are going to have a lot of elastic in them, so they're considered elastic arteries. So these elastic arteries have an important function, and it's that they propel blood onward in the pathway while the ventricles are relaxing. So as the blood is ejected from the heart into elastic arteries, their walls stretch and easily accommodates that surge of blood. But anything that stretches, remember, is going to recoil, and so that recoil is going to push the blood through that artery on into the next part of the pathway. So because these arteries conduct blood from the heart out to medium-sized arteries and then out to even smaller arteries, the elastic arteries are sometimes called the conducting arteries. Muscular arteries are called muscular because their tunica media contains more smooth muscle and fewer elastic fibers than the elastic arteries do. So the large amount of smooth muscle makes the walls of these arteries relatively thick, and they're capable of great constriction, so vasoconstriction and vasodilation, to adjust the rate of blood flows, and this of course is going to help to regulate blood pressure. So in large arteries, the thick tunica media can have as many as 40 or more layers of muscle cells lined like rings of a tree. In smaller arteries, there may be as few as 3 of those rings of muscle cells. So muscular arteries have a wide range of sizes, from sort of a pencil-sized femoral and axillary arteries to very, very small string-sized arteries that enter the organs. Because these arteries continue to branch and ultimately distribute blood to all the various organs, they're called distributing arteries. So the muscle, the ability of the muscle to be able to contract and maintain a partial state of contraction at all times is what we refer to as vascular tone. When you were learning muscle, remember muscular tone meant that state of partial contraction that helps muscles to maintain our body posture. Well, the same thing with the smooth muscle in these muscular arteries. So that partial state of contraction helps to stiffen the vessel wall and is important for maintaining blood pressure and also efficient blood flow through those arteries. Arterioles are small arteries, so they're microscopic vessels that regulate the flow of blood into the capillary beds throughout the body. The terminal end of the arteriole that feeds into the capillary bed is called the meta-arteriole, and we'll see this in just a minute when we look at capillaries. Arterioles play a key role in regulating blood flow from arteries into capillaries by regulating the resistance. Resistance is simply how much opposition the blood flow meets due to friction between the blood and the walls of the vessels, right? So anytime you've got something flowing through a tubular structure, there is resistance as that flow is flowing across or moving across the walls of the vessel. Because of this resistance, these vessels, the arterioles, are known as resistance vessels. In a blood vessel, resistance is due mainly to the friction between the blood and the inner wall of the vessels, like I mentioned. So the blood vessel diameter being smaller, the friction is greater, so there's more resistance. Contraction of the smooth muscle of an arteriole is going to cause vasoconstriction and increase resistance even more, and that can decrease the blood flow into capillaries supplied by that arteriole. On the other side of that, relaxation of the smooth muscle of an arteriole causes vasodilation. So there's a bigger diameter that decreases resistance and increases blood flow into those capillaries. So a change in the arteriole diameter by vasoconstriction or vasodilation can also affect blood pressure. Vasoconstriction of arterioles increases blood pressure. Vasodilation decreases blood pressure. Capillaries are the smallest of the blood vessels, and they form what you might think of as the U-turns in blood flow, because remember, arteries take blood away from the heart and it flows into arterioles and then into capillary beds, where the blood takes a U-turn and heads back to the heart into small venules and veins, and boom, right back where they started. So capillaries provide the U-turn. Some of the capillary beds and capillaries are so small that only a single erythrocyte at a time can pass through the lumen of these vessels. Sometimes they're even so small that the red blood cells sort of have to fold or deform themselves to squeeze through the tiniest of the capillaries. Capillaries form a very extensive network with approximately 20 billion capillaries making up the network of blood vessels in the human body. This network forms a very enormous surface area to make contact with the cells of the body. So in the images here you see a very small little capillary, and you can see that the erythrocytes are just squeezing through to get through that capillary bed and then into the venules and back into circulation back toward the heart. Another important thing about capillaries is because their walls are so thin, the capillaries allow for the exchange of nutrients, hormones, and waste products between the blood and the surrounding tissues around those capillary beds. So when we're looking at a capillary bed, the flow of blood from a meta-arteriole, okay, so we have an arteriole giving rise to a small meta-arteriole, is going to allow that blood to come into that capillary bed, and then as the blood passes through the capillary bed and also through this little shunt right here, it will then feed into a post-capillary venule, post-capillary, after the capillary, and then right back into small venules that are going to lead into larger veins. So this is sometimes referred to as microcirculation, micro meaning small, okay? So the microcirculation of the body. But the primary function of these capillaries is for exchange of material between the blood and the interstitial fluid of the tissues. So because of this, these small vessels are sometimes referred to as exchange vessels. Capillaries are found near almost every cell in the body, but their number in a particular tissue varies with the metabolic activity of that tissue. So tissues with a very high metabolic requirement, like muscles, which are always contracting, the brain, the liver, the kidneys, the nervous system, all of those are going to use a lot of oxygen and nutrients, so they have to have a much more extensive capillary network to be able to get that oxygenated and that nutrient-rich blood out to those tissues. Tissues that have a lower metabolic requirement, such as tendons and ligaments, contain fewer capillary beds. Makes sense, right? Because capillary walls are composed of only a single layer of those endothelial cells and a basement membrane, a substance can pass into and out of the blood with only going through one small, thin layer of cells to reach that interstitial fluid and cross back and forth from the tissues back into the blood. So when we're looking at the pathway of blood flowing into a capillary, notice that, as I mentioned a while ago, the blood's going to flow from an arteriole into a meta-arteriole, then into capillaries, and then into post-capillary venules. But at the junctions between the meta-arterioles and the capillaries, there are little rings of smooth muscle fibers that are called precapillary sphincters. What does a sphincter do? It opens and closes and regulates the passage of something, right? So these precapillary sphincters control the flow of blood through the capillary beds. So when the precapillary sphincters are open, then blood is going to flow easily through that capillary bed. But when the precapillary sphincters are closed or partially closed, the blood flow is going to stop or decrease through that capillary bed. So let's take a look at this. Here we've got tiny little precapillary sphincters, right? And if you look, if blood is coming into this meta-arteriole and these precapillary sphincters are open, then blood is going to flow out into all of the capillaries, and this just has a few little vessels out here. There would be hundreds of these. So blood's going to flow through all these capillaries out here, exchanging nutrients, dropping off oxygen, picking up CO2 in a very slow passage because it goes all through this capillary bed, and then the deoxygenated blood carrying that CO2 and less nutrients and such, right, is going to flow into the post-capillary venule and then back into circulation. So what if we want to restrict blood flow to that area? If these precapillary sphincters clamp down, then blood has nowhere to go except right through this vascular shunt. Okay, so the blood doesn't get routed out into these capillaries. So the nutrients, the oxygen, all of the good stuff stays in the blood and continues into circulation. Can you think of a situation where this might happen? I'm giving you a moment to think. Think about what happened when you studied thermoregulation back in Anatomy 1, and when you were cold, your instructor should have mentioned something about vasoconstriction in your peripheral tissues, vasoconstriction in your exterior tissues, right, in your peripheral tissues. So your fingers start turning blue and your toes start turning blue. Why did that happen? Well, that happened because you had vasoconstriction and clamping down of these precapillary sphincters out near your limbs, right? Well, why was that important? Well, we didn't want all that warm blood going out there through all of those capillaries and warming up the skin, warming up the limbs. We needed all that warm blood to go into the organs, right? Into your vital organs in your abdominal cavity and your thoracic cavity in your brain. So we clamp down those precapillary sphincters so that blood just rushes through and maintains all that oxygen, all those nutrients, and all that heat in your peripheral tissues. But in your vital organs, those precapillary sphincters are wide open. So all that blood then is able to flow through the capillaries of those vital organs and keep them supplied with oxygen so that you do have that blood supply to those organs and you can stay alive. You can survive without your fingers, but it's a little more difficult to survive without your heart and without your brain, right? This is also the same thing that happens when you have a fight or flight response. What happens, right? Your digestive system gets shut down, so you have vasoconstriction and precapillary sphincters clamping down in the digestive and urinary systems, so that blood rushes through without going through the capillaries, right? You don't want all that metabolic action with all those nutrients and oxygen going to your digestive and urinary systems. If you've got to fight a bear, the last thing you need to do is stop and pee and poop, right? So we shut down the blood supply or limit the blood supply to those tissues, and then we vasodilate and open those precapillary sphincters out into body tissues like your skeletal system, right, or your skeletal muscle, so that we have plenty of oxygen and nutrients going out so we can make a lot of ATP because we're going to have to fight or flight. There are three types of capillaries found in the human body: continuous capillaries, fenestrated capillaries, and sinusoids or sinusoidal capillaries. Most of our capillaries are continuous capillaries. We find these in the skin and the muscles, and they have a continuous lining of endothelial cells that forms a continuous tube that's interrupted only by just a few intracellular clefts or gaps between the neighboring endothelial cells. So, as I mentioned, we find these in the muscle tissue and the skin, but also in the central nervous system and in some areas of the lungs as well. So these allow for the passage of very small solutes and fluids. These are not present in the brain, though. However, we have something known as the blood-brain barrier that you learned about when you studied the nervous system back in Anatomy 1. So the capillaries that are found in the brain allow only a few things to cross their walls. So most areas of the brain contain a specialized continuous capillary where their very, very tight junctions basically seal all of these capillaries together, so the cells that make up the capillary walls. So it results in sort of a blockade to movement of materials into and out of the brain capillaries, and that makes up the blood-brain barrier. But there are areas of the brain, such as the hypothalamus and the pituitary gland, that have a little bit more free exchange across the walls of those capillaries. Your fenestrated capillaries have endothelial cells that have fenestrations or small holes. So fenestra means window. So fenestrated capillaries are found in the kidneys, in the villi of the small intestine, the choroid plexuses of the ventricles in the brain, in the ciliary processes of the eye, and in most of the glands. So these are much more permeable, and they allow small solutes to pass. The sinusoidal capillaries are sometimes just called sinusoids or wider, and the endothelial cells have really large fenestrations or big windows. You can think of these like sliding glass doors. So they have very large intracellular clefts also. So the large windows and the large clefts allow proteins and some blood cells to pass from the tissues into the bloodstream and from the bloodstream into the tissues. For example, newly formed blood cells enter the bloodstream through the sinusoids of red bone marrow capillaries. Sinusoids in the liver contain phagocytic cells that remove bacteria and other debris from the blood. The spleen, the anterior pituitary, parathyroid gland, and adrenal glands also have these very large open sinusoidal capillaries. Venules and veins have thin walls that don't really maintain their shape well. Venules drain capillary blood and then return that blood flow back toward the heart as they merge and become larger and larger and larger veins. The venules that initially receive the blood from the capillaries, as we saw a few minutes ago, are called post-capillary venules. They are the smallest of the venules, and they, of course, are going to get that blood from the capillaries and then merge and form larger and larger veins. So just a reminder of some of the things that we've seen with veins. The thinner walls, so thinner tunica externa, very, very thin tunica media, and then just the normal lining of the endothelial lining, the tunica interna. But I want to go ahead and point this valve out right here because we will be talking about the importance of this valve when we're looking at blood flowing back to the heart. So only certain veins have valves, and the valves are oriented so that blood can push upward back toward the heart, but when it comes back down, it smacks against those valves and pushes them closed, very much like a swinging door going into a saloon that will only open in one direction. The other image you see up here is a microscope image of a cross-section through a vein, and you notice how thin the walls are. So a lot of times when you're looking at a slide, if there is an artery and a vein both, and you're wondering which is which, the vein will usually appear much more collapsed. Whether it's bigger in diameter or smaller in diameter, the vein will usually be the one collapsed, and you should notice a much thicker wall when you look at a cross-section of an artery. So again, veins are formed when you have the merger of several small venules. And although veins are composed of the same three layers as arteries, the relative thickness of the layers is different, as we saw in the last image. The tunica interna of the veins is thinner, but not a lot. The tunica media is much thinner in the arteries, with relatively little smooth muscle and elastic fibers. The tunica externa of veins tends to be the thickest layer, and of course it consists of the usual, the collagen and the elastic fibers. The lumen of the vein is larger than that of a comparable artery in most cases, and as I mentioned on the microscope image a while ago, the veins often appear collapsed or flattened. When you look at a microscope slide, you're looking at a section through those veins. The average blood pressure in veins is considerably lower than it is in arteries. Remember, arteries are taking blood away from the heart, so the arteries receive that blood as the ventricles contract, and it's under high pressure, and that pressure stays high, getting less and less, but still high as it passes further and farther and farther away from the heart. But the difference in pressure, of course, in these veins can be noticed if you cut a blood vessel. So let's say you get an injury and blood cuts through a vein. It's going to bleed, but it's very slow flow. If you cut an artery, you've got blood spurting everywhere. So when you're watching one of the medical shows on television and they're doing surgery and you see blood spurting off the operating table, of course that's a little dramatic, but it does happen occasionally, and that tells you that an artery has been cut. You're going to lose blood very quickly. Most of the structural differences between arteries and veins reflects this pressure difference. The walls are not as strong as those of the arteries, therefore they are not capable of withstanding as much pressure. Many of the veins, especially those that we find in the limbs, contain the valves that I showed you a few minutes ago. So those folds of the tunica interna that form those flap-like cusps with the flaps pointing upward toward the heart. So these valves aid in the venous return by preventing backflow, as we saw. So rather than looking at the little cartoony image we had in the last one, here you can actually see a valve in a vein. So it is just an infolding or an extension of that tunica interna. A vascular sinus, also called a venous sinus, is a vein with a very, very thin endothelial wall that doesn't have smooth muscle that can alter its diameter. For example, the dural venous sinuses are supported by the dura mater, and they convey deoxygenated blood from the brain back down to the heart. Another example is the vascular sinus, that is the coronary sinus that we saw, that collects the deoxygenated blood from the walls of the myocardium of the heart itself and returns that back into the right atrium. So let's look just briefly at a homeostatic imbalance associated with veins, and these are what we call varicose veins. So leaky valves can cause veins to become dilated or larger, and in some cases they look sort of twisted in their appearance. So this is a condition that we call varicose veins. It can happen in any part of the body, but it's most common in the esophagus and the anal canal, and in the superficial valve or superficial veins of the lower limb. The ones in the lower limb can range from cosmetic problems to serious medical conditions if it causes changes in the blood flow pattern. A valvular defect may be caused by congenital defects, or it can be mechanical stress such as prolonged standing or the extra weight and pressure associated with pregnancy, or vessels wearing out as we age. The leaking valves allow backflow of blood from the deep veins to the less efficient superficial veins, and that's where the blood pools. So this creates pressure that distends the vein and allows fluid to leak into the surrounding tissues. As a result, the affected vein and the tissue around it can become inflamed and very tender. Veins close to the surface of the leg, especially the great saphenous vein, are very susceptible to varicosities, varicose veins. Deep veins, not so much so, because the surrounding skeletal muscle prevents their walls from stretching so much, so more likely to develop these in the more superficial veins. Varicose veins in the anal canal are referred to as hemorrhoids. Esophageal varicose veins result from dilated vessels in the wall of the lower part of the esophagus and sometimes the upper part of the stomach, and can lead to bleeding and life-threatening conditions as a result of this. So several treatment options exist for varicose veins of the lower limbs. Sometimes it's simply elastic stockings that we call support hose can be used if the varicose veins are mild, and for people that other options are not recommended. Perhaps they have other health or cardiovascular issues. Scleropathy is a treatment that involves injection of a solution into the varicose veins, and that solution damages the tunica interna by inducing a superficial thrombophlebitis. Boy, that word didn't come out right. Thrombophlebitis. I give up. Inflammation that involves a blood clot. Of course, that's going to cause the immune system to try to come and fix that area. So healing of that damaged part causes a scar formation, and that typically occludes or shuts down that vein. There are some radiofrequency therapy that uses the energy to heat up the vein and close off the varicose veins. Lasers can be used to destroy the veins. There's also a surgical procedure called stripping where the veins can be removed. So in this procedure, a flexible wire is threaded through the vein and then pulled back out, and when it's pulled back out, it basically just brings the vein with it. Yeah, I think I'd need lots of pain medication to have that one done. How about you? An anastomosis is where you have the union of two or more arteries supplying the same area of the body. So this provides an alternate route for blood flow. So if one artery is blocked and blood can't get to that particular area of the body, necrosis or tissue death would occur. However, in areas where it's critical that we do have blood flow, such as within the brain, we have alternate pathways, and that's basically what an anastomosis is, to make sure that blood flow continues to and from a particular tissue. You find a lot of arterial anastomosis in areas that are, of course, vital to the life, such as the brain, the heart, and the abdominal organs. You also find these, though, in the joints, because think about what our major joints of the body are doing. we're constantly flexing and extending those joints. So if you're sitting for a long period of time and your legs are bent, then you may be obstructing the flow of blood to the lower part of your limbs, the lower part of your legs. So there are alternate pathways rather than the major pathway that you might be obstructing. So these alternate pathways make sure that blood flow does occur. You do have numerous venous anastomoses, and one that you can see pretty readily is to look at the blood vessel pattern on the back of your hand. And you should notice that you have several superficial veins there, and between many of those veins you will have tiny little veins that connect two or more of those major pathways. When you're at rest, about 64% of your blood is in your systemic veins and venules. And because they contain such a large percentage of your blood volume, they function as blood reservoirs. So a reservoir is like where you store something until you need it, right? So we can take blood and easily divert it quickly from these areas if need arises. So if you have increased muscular activity, then the cardiovascular system in the brainstem sends a large number of sympathetic impulses to the veins. The result is venoconstriction, or constriction of the veins, and that means, of course, some of the blood is going to be pushed out of those and back into normal blood flow to get out to those skeletal muscles where we're using more ATP. We need more blood. A similar mechanism operates in the case of a hemorrhage. So when blood volume is lost, blood pressure is going to decrease, and in that case, venoconstriction helps to counteract the drop in blood pressure, because when those veins vasoconstrict, then or venoconstrict as we typically call it, then of course the pressure increases in those, pushes more of the blood out into those arteries that are supplying major tissues. So the blood vessels, the veins, and the venules that are the principal blood reservoirs are the veins of the abdominal organs, especially the liver and the spleen, and of course the veins of the skin, where we can divert that blood quickly to areas that need it.